allowing me this opportunity. So yes, I'm Amir Ohad. I'm from Yasmin's lab in Tel Aviv, and it's definitely hard to follow your boss in the talk, so I'll do my best. And so you'll see that there might be some repetitions. So in this BBC video, we see the climbing plant moving in these large circulatory motions we call circumnutations. At first, there is this small attachment to the leaf on the right, and then it eventually stops trying to twine on this leaf, and it eventually manages to clasp onto the much more stable support on the left, this sturdy branch. And so our basic question going into this project was, how do plants assess mechanical stability of supports, assuming that twining plants are unable to sustain themselves mechanically on their own? They have to attach to some external support. So it makes sense in an evolutionary way that they would not waste energy in a sense on supports which are not stable. So we want to see how they can do this kind of assessment. Stability is typically tested by trying to apply some force to what you're trying to test the stability of, and at some point it might fail. And so we want to see if we can apply the same kind of logic to climbing plants. So our hypothesis is that the twining plants use circumnutation to assess the mechanical stability of potential supports. And we do this by observing the mechanics of the dynamics of motion and force of contact during these circumnutations with a potential support. And we see that there are other animals that do this kind of assessment of mechanical stability, such as cats and mice with their whiskers, this thing called whisking, which allows them to sense external objects with this kind of flexible motion. So our setup is basically a pendulum. So you have this kind of, so this very hard to see black line is a plastic rod, and it's connected with a hinge to this part here. I have two cameras tracking the movement of this rod, so one from above and one from the side. And basically the circumnutating organ, which is usually 30 to 40 centimeters high, and I will measure the circumnutation duration before the experiment begins to have a notion of the timescales for this specific plant. And then I'll allow it to impact onto the support, and I can measure the angle generated by this contact, and from this I can extract the forces of the plant applying during circumnutation. So here you can see the side view camera, and in this case, eventually you'll see that it will twine onto the support. And you can, so the way I define twining is by the angle of this part of the shoot relative to the vertical. So once it goes towards the vertical, then I consider it to be a twining event. We have different kinds of events, which we call slipping events. So this is the top view, and you see that at some point the plant will slip off of the support. So these are the two main categorizations we have for these events. And this is an example force trajectory for the second event. You see that the force rises in this kind of curved fashion, and eventually it drops off to zero after the slip event. Okay, so now we try to see if we can understand where the forces are coming from, if we can simulate these forces in a theoretical fashion. So Amir, who did his PhD at our lab, who's now working at Cambridge, did these really nice simulations based on the Elastica software and properties that we measured in experiments. So we gave the experimental Young modulus of the plant, so the stiffness and the radius of the organs that we were dealing with, to generate these simulations. And you can see he can measure the forces generated with the simulated support. And so now we can see if these simulated forces actually match the forces that we measure in experiment. As you can see here, these are the raw trajectories of the forces, and this is the time in minutes. and you have a lot of variation of the kinds of forces that we measure. However, if we fit each individual force up to the point of either twining or slipping, then if we normalize them and fit it by fitting them to some sine function, then we get a very nice collapse of the force trajectories onto the simulated force trajectory. So this shows us that we can quite comfortably understand the origin of the forces generated before the twining event or before the slipping event by understanding them as generated by just pure circumnutation motions. But we have a problem, which I'll get to in a bit. So the second thing we understand now that we know that the circumnutations are generating the, at least qualitatively, the force, we want to understand what will separate between the twining events and the slipping events. So the first thing we notice is that there's a geometric effect. So when we have contact that is very close to the tip of the plant, so this is the support from the top view, and here you have the distance to the tip of the organ. And when this distance is very small, so if the contact was close to the tip here, you'll see that most of the events in orange tend to be slipping events. However, once we increase the distance, so if the contact is closer to the base, then the events tend to be of a more twining, tend to result in twining. So this is very geometric, not very complicated or interesting, but it's there. And the second thing we notice is that there's a temporal effect. So twining events seem to occur only after about half of a circumnutation duration. So T is the contact duration, and TCN is the circumnutation period, individual for each plant. And we see that there's a very sharp cutoff around half of a circumnutation period. So almost no twining before this time. And if contact is maintained for half a circumnutation duration, the probability for twining is very, very high. So this was quite striking, and we tried to understand now where this is coming from. So the first thing we notice regarding these timescales after this was that there seems to be some mismatch between the simulations and the measured trajectories. So I did show that they fit very nicely before, but that was when I normalized the timescale according to a fitted sine function. So there was one constant time period that I'm normalizing the force by. However, if I normalize the forces according to the natural circumnutation duration, then I see that there seems to be some stretching out of these force trajectories. So they don't fit the quarter times the quarter circumnutation period that we would expect from a constant circumnutation rate. So there seems to be some sort of stretching out. So maybe the contact, this made me think that maybe the contact is sort of increasing the effective circumnutation period. So to try to quantify this, I performed another experiment where you have contact in this position, and if contact was not occurring at that point, so let's say I start a clock at this point, and I wait for a certain amount of time t, then this would be the expected angle I would have. So theta expected is after, say, t duration of contact. However, what we see in practice is if the slipping event occurs after this time, then the experimental angle is actually smaller than what we would expect. And it seems, okay, so I would do this intentionally. I would allow contact for a certain period of time t, and then force a slipping event by, say, moving the support. And then I would see with the resulting angle. And so we see that if for increasing contact durations, again normalized by the individual circumnutation period, that this delta theta, so the difference between the expected angle and the measured angle, increases quite linearly with the contact duration. So the shift in the circumnutation period, or the internal clock, is dependent on the time of contact. And so next we wondered, we keep seeing this circumnutation period pop up in all of these measurements. So we're thinking, can if I modify the circumnutation rate, will this affect the resulting dynamics? So how can we change the circumnutation rate. So I was talking yesterday with Dr. Hollander and Dr. Magari also about this question, whether I can use mutants or by drying up the plants. So these are things I haven't tried yet. But I was thinking from a more mechanical way, I can actually use a motor. So I can change the motor. I can change the rotation rate of the plant by having a motor stage either move against the circumnutation direction, and this will decrease the effective rotation, or I can turn it in the other direction, and then the rotation rate will increase. And want to see how this will affect the resulting twining timing rates. Okay, so this is an event where I'm increasing the circumnutation, the effective circumnutation rate, and this twining occurs at around, it's about 35 minutes, where the general time would be over 40 minutes. So I'm already increasing the rate of the twining. And have we seen twining events occur for even shorter periods for faster rotation? So that's one time. And the second one is if I slow down the circumnutation, so you can see the plant is being moved clockwise with the motor, and the circumnutations are always in beans, and these beans are always counterclockwise. You can see contact is maintained for a very long time, yeah, and there is no twining occurring. So this is the other extreme, where you have contact being maintained for a very, very long period of time relative to the natural circumnutation rate, but no twining is occurring. And so when we plot this relative to our experiments, so the constant or the natural circumnutation rates are all of these black dots. And you can see that the trend continues if I change the rotation rate with the motor in this very nice decaying fashion. So the fit is only to guide the eye. It's not based on a theoretical model. But we can extrapolate this in both directions and see that with very high rotation rates, we'll get very, very short twining rates. And conversely, if I decrease the rotation rate to close to zero, then the twining time will be practically indefinite. So I haven't reached those limits, but that's what we would get for a simple extrapolation. And so to summarize, we see that the twine probability is governed by both the geometric factor and the contact duration, and that simulations show that we can generate the force trajectories pretty efficiently, at least in a qualitative sense. And so plants appear to be using circumnutation to apply force to these candidate supports. And then in the second subject, that we can see that contact is decreasing the circumnutation rate in a linear fashion, or appears to be a linear fashion, and that if we modify the circumnutation rate, then we can affect the twining initiation time accordingly. So twining initiation is dependent on some applied force, but prolonged contact is not sufficient. So as you see in the last video, we can maintain contact with the support for a very long time and not trigger twining. So this raises some very interesting questions. And with that, I would like to thank you for your attention, Yasmin and Amir, for all the work, and the rest of my lab members, and I'll be glad to try to answer any questions. Thank you.
Thank you. Questions? Yeah, you can start, and then
does it work? Yeah. So the contact, isn't it like that when you rotate the plant, the contact side will basically rotate, so you confuse the system and it cannot initiate the twining because you stimulate the stem from one side and you rotate it, so you basically confuse it with the contact side.
So I do still see twining occurring, even for larger, even for longer periods. So it does eventually happen at slow rotation rates. It's not that it can't do it. Just the accumulation of this signal is much slower. So it's not that it's not possible.
But in a movie, it looked like that when you slow it down, it's really like rotating around the.
So okay, so in general, these circumnutations, the plant, the organ is constantly turning about itself. Like this is part of the natural circumnutation movement. So the plant is continuously doing this rolling kind of. So there's no a hinge, right? The plant is, there's no hinge. It's rolling about its own stem. This is the circumnutation motion. And so this is just an increased, a more accentuated version of this. I think, I hope that answers it.
Okay, yeah, last questions because we are a bit out of time.
Okay, thanks for the talk. That's super interesting. You show in your conclusion slide that basically what happens is the plant puts forces on what is going to be the next support, and it depends on the contact time. So what I wonder is, does the plant also change the forces it applies on the contact surface to maybe measure actually if it is a good support?
So as you saw in, so I showed the force trajectories. The force trajectories are very different. Like there's a large variation in the actual force applied, but twining can be triggered for a large variant of these. So it doesn't appear to be the maximal force that is actually the relevant signal. But you're right that like a very, very small signal will probably not be enough. So a very, very low force will not be sufficient. So there is some minimal force, but I'm not sure that a maximal force is the correct signal to be looking at.
Okay, thanks.
Okay, okay, thank you. Let's thank Amir again. So the next speaker is Pawan Jewaria from the National Institute of Plant Genome Research in New Delhi.